Chapter 8 in our course, Chapter 24 in the textbook, covers biomolecules. And that means we're going to need to look at organic chemistry and biochemistry. Organic chemistry is the covalent chemistry of carbon. And we already know from Lewis structures that carbon always makes 4 bonds. That's going to be important. Biochemistry is the chemistry of living organisms. And that turns out to be mostly the organic chemistry of carbon in combination with hydrogen, oxygen, nitrogen, phosphorus, and sulfur. One really important thing about biochemistry is that shape is very, very important, which means thinking about our molecules as two-dimensional representations on paper is probably not going to be good enough. We're going to need to think about molecules in 3 dimensions. Okay, so we have this knowledge already that a Lewis structure is an electron dot structure, and atoms combine by covalent bonding, by sharing their valence electrons, and in fact only the unpaired valence electrons, until every atom in the new molecule has 8 valence electrons. So here's some hydrogens, each with 1 electron. Here's a carbon with 4 valence electrons. They come together. The hydrogen has a covalency of 2, which is all it needs because it's in period 1, and the carbon has a covalency of 8. Writing those dots out every single time gets a little tedious, so we're going to replace Lewis structures with Kekule structures. And in a Kekule structure, we now represent 2 electrons shared between 2 bonds, a covalent bond, as just a straight line. Covalent bond is 2 electrons shared between 2 atoms. So carbon has 4 bonds. Hydrogen only has 1. Carbon is always going to have 4 bonds. Hydrogen is always going to have 1. Oxygen is always going to have 2. Nitrogen is always going to have 3. This is enough to get us started. Hydrocarbons are molecules that contain only carbon and hydrogen. Now, there are a couple of different subclasses of hydrocarbon. We're going to look at the simplest first. They're called alkanes. Alkanes are what we would consider saturated hydrocarbons in that they contain only single bonds. So a central carbon with 4 hydrogens around it gives us a chemical formula of CH4. Sometimes this is called a condensed formula, and this is called a structural formula or a Kekule structure. And this is given name of methane. The first part of the word tells us the number of carbons. The second part of the word tells us what family this molecule belongs to. Anything that is an alkane, contains only single bonds, ends in ane. Meth means 1 carbon. Eth means 2 carbons. So a 2-carbon saturated hydrocarbon, ethane, has the 2 carbons attached to each other and then 6 hydrogens around it. C2H6 is ethane. C3H8 is propane. For 4 carbons we would have butane, and then pentane, hexane for 6, heptane, and then octane for 8. Now, there is something that becomes apparent. Every carbon has to have 4 bonds. So this carbon is holding on to those 2 hydrogens. This carbon is holding on to those 2 hydrogens. This carbon is holding on to those 2 hydrogens. And then we have the 2 extra at the end. So alkanes have a general formula of CnH2n plus 2. However many carbons you have, double that number for hydrogens and then add 2 because those will be the hydrogens at the end. Alkenes are a form of hydrocarbon that contain a carbon-to-carbon double bond. Because alkenes contain less hydrogen than alkanes do, alkenes are called unsaturated molecules. Our simplest alkene is ethene. Eth meaning two carbons, ene meaning containing a carbon-carbon double bond. Two carbons stuck together with a double bond. Each carbon has 4 bonds. Each of the hydrogens has 1 bond. I have a formula of C2H4. Propene, 3 carbons. In a chain, there is a double bond, and each of those carbons has 4 bonds, therefore 8 electrons. And then when I get to butene, something happens. At butene, I could put the double bond here between carbons number 1 and 2. We're going to number our carbons in a molecule, 1, 2, 3, 4. And I've chosen to put the double bond here. But that's not the only place that I could put it. I could put the double bond between carbons 2 and 3, which means that these two molecules share the same chemical formula but have a different shape. These are structural isomers. This one is called 1-butene and this one is called 2-butene. In naming an alkene isomer, we determine where the double bond goes, and then, so here it's between carbon number 1 and carbon number 2, and we include in the name the lowest number of those 2 carbons. I don't need to tell you that it's between carbon number 1 and carbon number 2, because if I tell you that it's from carbon number 1, you know that it's going to carbon number 2. So just use the lowest number. Let's look at 3 different types of isomerism. The first is structural isomers. So here I have butane. Butane is 4 carbons in a chain, meth, eth, prop, but. Ane tells me only single bonds, and we count the longest chain. So this is 4 carbons in a chain, that's butane, C4H10. But if I took that methyl, 1 carbon group, methyl, off the end and reattached it somewhere else, so I'm breaking a bond, moving a fragment, and then reattaching somewhere else. This is a change in configuration. Configuration is the bonding motif. It's the accounting of where do all of the bonds go. I've made a structural isomer, and now my longest chain is 1, 2, 3 carbons long. And because we're thinking about a 3-dimensional molecule, don't get into the habit of reading them from left to right. You could read this 1, 2, 3, still 3 carbon chain. 1, 2, 3. Any way that I count it, my longest chain is 3 carbons long. So this is now a propane of some description. Still only single bonds, so still an ane. Prop, longest chain is 3 carbons long. But now there is a 1 carbon fragment or substituent stuck onto that molecule somewhere, and it is at the 2 position. 1, 2, 3, propane with the meth stuck at the 2. So this gets to be 2-methyl propane. A consequence of adding a carbon-carbon double bond into a molecule is that double bonds are planar. They're rigid, they're flat, and this part of the molecule is subject to a special type of isomerism called geometric isomerism. This is butene. We'd seen butene earlier, and we'd decided that there were 2 types of butene, 1-butene and 2-butene. But there's slightly more. Because this part of the molecule is flat and rigid, It means that having the methyls be on the same side of that double bond is different shape-wise than having them on opposite sides of the double bond. And so now I have two new geometric isomers. This is the cis 2-butene, where the cis isomer means on the same side of the double bond. And this is the trans 2-butene, where the trans means on opposite sides. And again, in chemistry, and specifically in biochemistry, shape is absolutely everything. And it could be that a molecule that is this shape is recognized in biology, but a very simple isomer of it would not be recognized. Might have some biological function, might not have that function. Lastly, we'll look at optical isomers. Optical isomers are a special type of molecule that bend plane polarized light either to the left or to the right. You might be aware that water refracts light. If you put a stick into a pond, you'll see that stick bend. Well, it turns out that some solutions of molecules will bend light to the right and some will bend it to the left. And this comes about when the molecule contains a chirality center. Chirality. Chirality is when a tetrahedral carbon is bonded to 4 different groups. And that can start to appear when our molecules are still fairly small and easy to draw. So this is 2-butanol. It is an alcohol. It has 4 carbons in a chain. And at carbon number 2, there is an OH, an alcohol group, which means that red carbon is attached to 4 different things. It's attached to a methyl, a 1-carbon fragment, an ethyl, a 2-carbon fragment, a hydrogen, and then a hydroxyl, the alcohol group itself. So when we write this out in 3 dimensions, because it's not really something that we see in 2 dimensions, this is a 3-dimensional type of isomerism. Remember, this type of drawing tells us that the wedged bond is pointing out towards us, the hatched bond is pointing away from us, and any bond that is still just a line, that's in the plane of the paper. This is a mirror. Everything that's closest to the mirror is the first thing that gets reflected. Oftentimes, the mirror image of an object is a perfect replica of that object. But in the case that you have an asymmetry because of a chirality center, this is now a different isomer than that. These are non-superimposable on each other. They are different. So this now gets to be called R2-butanol. This is the right-handed isomer, where the R is rectus, which stands for right-handed. This gets to be the S isomer, which is the left-handed isomer, sinister for left-handed. And this tiny little subtle change in configuration here, in how the atoms are attached, has tremendous importance in biochemistry, because remember, shape is absolutely everything. And there are hundreds of cases that you can go out and find of 2 molecules that have the same name, they have the same structure, but they have a chirality center, and so they have this subtle difference. And one of them is biologically active, and then the other one is not. If you're lucky, the other one is not. Sometimes This one does something useful and has been included as a drug, and then this one has horrible side effects. The big one that comes to mind is thalidomide. Thalidomide was a drug that was given to pregnant women in the 60s to help prevent morning sickness and caused horrific birth defects. Turns out one of the isomers was perfectly harmless, did its job. The other one caused the birth defects. Lots of biological molecules have chirality centers in them and have different activities based on which particular isomer you're looking at. Most molecules contain a functional group. Functional group contains an electronegative atom and is the site at which future reactions are going to occur inside this molecule. Let's run through a few simple ones. Alcohols. Alcohols have a carbon to oxygen to hydrogen bonding set. So this is one carbon alcohol, methanol. Amines, remember nitrogen always makes 3 bonds, oxygen always makes 2, nitrogen always makes 3 bonds, contain a carbon to nitrogen set of single bonds. So a 2 carbon fragment attached to a nitrogen is an ethyl amine. Aldehydes contain a carbon that is doubly bonded to an oxygen, but also singly bonded to a hydrogen. Then the rest of the molecule just becomes the first part of the name. So 1, 2, 3, meth, eth, prop. Oh, I didn't include the carbon for the molecule itself. This is butanal for an aldehyde. Ketones also have a carbon to oxygen double bond, but this carbon to oxygen double bond is in the middle of a carbon chain. Aldehydes, the carbonyl group is at the end of a chain. Ketones, it's in the middle. So this gets to be butanone. One for ketones, al for aldehydes. These 3 functional groups are all based around the carboxylic acid. These are what we would call carboxylic acid derivatives. Carboxylic acid itself has a chain, and then this carbon is doubly bonded to an oxygen, this carbonyl, and then bonded to an alcohol group. So this carbon is bonded to oxygen 3 separate times. And we just name the number of carbons that are in the chain, 1, 2, F, and then anoic acid. Esters are just like the carboxylic acids that they came from, but instead of having an OH group, an alcohol, they have an O to carbon single bond. So a carbon to oxygen to carbon set of single bonds is called an ether. If a carboxylic acid is a carbonyl alcohol, then an ester is a carbonyl ether. And we name this as a substituent, that's the methyl, and then we name the long chain ethanoate. I think I want you to recognize esters, not particularly need you to name them. Just recognize that they are different than carboxylic acids because of the attachment of this carbon. Amides, just like carboxylic acids, but where this alcohol group, oxygen only makes 2 bonds, this amine group makes 3 bonds. So I have a chain, I have a carbon to oxygen double bond, a carbonyl, and then I have an amine group, And a carbonyl and an amine attached to the same carbon is an amide. Longest chain is only 2 carbons long, so this is ethyl amide. Let's look at some biomolecules. First, we'll look at carbohydrates. Carbohydrates are sugars. Sugars arise from photosynthesis, where green plants take sunlight and combine that with carbon dioxide and water to make this, a carbohydrate. And they also give us 6 molecules of oxygen. This process takes energy in the form of sunlight. We take the sugars from plants and the oxygen that they give us. We breathe in the oxygen and we ingest the carbohydrate and we metabolize it and we get that energy back out. So sugars are a source of energy for us. And the sugar that we're most interested in, there are lots of different sugars. If you hear a molecule end in ose, if its name ends in OSE, it's a sugar, and there are lots of them. But far away the most important one is glucose, and glucose has formula C6H12O6. So if you think for just a second, for each of those 6 carbons, there are 6 waters. Carbohydrate. This is the structure of glucose. And it's a little bit awkward, but you can see it's got lots of alcohol groups. It also has an aldehyde. Each of these carbons in the middle that don't have a label, because this is what we call a Fischer projection, don't need to worry about that. Each of those unlabeled carbons has 4 different groups around it. So this molecule is highly chiral. Its shape is subject to change by changing any one of those positions. So this is something very, very important. It turns out that this aldehyde and that alcohol like to cyclize, and so sugars are often drawn in one of these 2 forms, where they're a hexagon. This is called a Haworth projection, and then this is often called a chair because it vaguely resembles a chair. And you can see that it is a ring that contains an ether, carbon to oxygen to carbon bond, and lots of alcohols on the outside. The little sugar molecules themselves are going to be called monosaccharides, and they're involved in a polymerization reaction, which is actually a condensation, that 2 molecules of sugar are going to lose a molecule of water and condense together to join together. And a monosaccharide, like glucose, can combine together to make a disaccharide, 2 sugar molecules stuck together. This one is lactose, which would then continue to polymerize. And we go from being a monosaccharide through a disaccharide to being a polysaccharide, things that are known as complex carbohydrates. Simple sugars, complex carbohydrates. And this one is cellulose. Cellulose is found in fiber because it turns out that sugar is used for 2 different things. It is used as structure by plants. This cellulose right here would be the structural component in grass and other plants that are like it. But humans don't digest cellulose. So if the molecules of glucose attach to each other in this particular way, they make a molecule that we can't digest. However, if they attach to each other with just a simple change, and instead of that bond going up, it were to go down and then back up, my glucose makes a maltose, and when all the glucoses are attached together, I make starch. And starch, you find that in potatoes. This is globular and is edible, can be metabolized by humans. So very important, again, shape being the important factor between two completely different functions in the plant world. The next biomolecule that we'll look at are lipids. Lipids are fats, and fats are a much misunderstood, much maligned molecule, where carbohydrates had two purposes, structure and energy storage. Fats have multiple purposes. They're used as shock absorbers, like right here, so that every time you fall down, you don't break the bones in your hand. They're used for buoyancy, they're used for insulation, they're used for secondary sex characteristics, and they're used for long-term energy storage. And lipids themselves are a condensation product between glycerol, which is this molecule here, 3 alcohols, and 3 fatty acids. And a fatty acid is a carboxylic acid, but with a long chain. So this squiggly line just tells me that's something that I haven't completely defined, but it's a long chain of carbons. They all turn out to be even numbers. Nature does a couple of things, that it only uses certain types of molecule. All fatty acids are even numbers. Fatty acids can be long chains that are saturated, containing only single bonds, completely surrounded with hydrogen. Or they could be unsaturated. You've probably heard that animal fat is saturated fat, contains only single bonds. Vegetable fat or vegetable oil contains unsaturated fat, which contains double bonds. Again, nature does something for us. It only uses cis double bonds. It does not use trans double bonds, which is why having trans fat in our diet is something we should be concerned about, because it's not something that our bodies recognize and are capable of metabolizing correctly. They've never seen trans fats before this most recent generation. So I'm going to go in here and I'm going to remove 3 molecules of water. That's a condensation. I'm going to make a molecule that looks like this. Okay, so this is the fat part. These are its components. And one thing that has already come up for us during the term was a discussion about detergents and soap. And if you want to break fat down and get these fatty acids back out again, because they are useful, because this end right here is polar, but that end right there is non-polar. Fatty acids are the source of soaps for us that have a salt of the polar end at one end, which is going to be soluble in water, and then the long chain that is soluble in non-polar oils, so that it can remove the oil into the aqueous layer. Next, we'll look at proteins. Proteins are the condensation polymer of amino acids. So we're going to take some amino acids, we're going to lose some water, and we're going to build something called a polypeptide, which as you continue to add more and more amino acid residues to it, becomes known as a protein. Now, amino acids contain 2 functional groups. This is an amine, and this is a carboxylic acid. I just erased the oxygen. If I take 2 amino acids and put them close to each other, I get the possibility of removing a molecule of water and therefore attaching the carbon in this molecule to the nitrogen in that molecule, making an amide. That amide in biochemistry terms is called a peptide bond. And this is how we turn individual amino acids into peptides. Now, if you read the book, you'll find that the amino acids don't completely exist in a form quite like this, but it's the easiest way to draw them out, and it's the simplest explanation. So each of our amino acids, and there are about 20 amino acids that are naturally occurring. That number, surprisingly, even now is up for debate, but we're going to say that there are 20 of them. I have an amine attached to this carbon. I have a carboxylic acid attached to the same carbon. I have a hydrogen, and then I have something else. The something else is a substituent that hangs off the amino acid and is known as the R group. This side chain is going to become increasingly more important as the size of the protein grows. Because if this side chain has an electronegative atom, then the side chain will be polar, and polar molecules like to interact with water. And we make a hydrophilic environment, a part of the protein that likes to interact with water, probably going to be on the outside. If we have just non-polar groups here, just regular carbon substituents, then I build a hydrophobic environment, one that doesn't like water, is non-polar, and then the molecule folds around on itself. And we read in the book that there are 4 types of protein structure: primary, secondary, tertiary, and quaternary, telling us that the R groups enforce a environment where like wants to be next to other things that it is like, and the non-polar groups are going to congregate together and the polar groups are going to congregate together. And that gives us shapes of proteins, and proteins can have 250,000 atoms in them. Here's a table of common amino acids sorted into the 2 major categories of non-polar, hydrophobic, where you can see that the highlighted side groups right here, those are the R groups, and then the polar, hydrophilic side groups right here. Each amino acid, the side group changes the name of the amino acid. The amino acid gets a name, it gets a 3-letter symbol, and it also gets a 1-letter symbol, because when we write out protein structures and we have perhaps hundreds of thousands of atoms in a protein, using a single letter to describe glycine is more effective than using 3 letters. Our remaining group of biomolecules are nucleic acids. This is a nucleotide. It contains a phosphate group, a sugar. This is based on ribose, which is a 5-carbon sugar that's missing an oxygen, so this is deoxyribose, and then a nitrogen-containing base. There are 2 different types of nitrogen-containing base used in nucleic acids. They are either pyrimidines, known as thymine and cytosine, which are 6-membered rings that contain 2 nitrogens, or purines, like adenine or guanine, which are 2-ring molecules that contain 4 nitrogens. Now, it turns out that hydrogen bonds can exist between adenine and thymine, and also between cytosine and guanine. And a hydrogen bond arises when the lone pair of electrons on an electronegative atom, like this oxygen, lines up in proximity to a hydrogen on another molecule that is attached to an electronegative atom. So this hydrogen is feeling electron withdrawal, and this oxygen has electrons that it would like to share. And this generates a very weak electrostatic attraction. And when you put something that is capable of giving and receiving a hydrogen bond in close proximity, those molecules stick to each other. A and T, adenine and thymine, stick together with 2 hydrogen bonds, and cytosine and guanine stick together with 3 hydrogen bonds. This process of sticking molecules together from individual nucleotides is called base pairing, and it's named after Crick and Watson, the scientists who discovered the structure of DNA. Okay, so individual nucleotides, here's a nucleotide that contains adenine, the deoxyribose, and a phosphate, can polymerize. That the alcohol group out of this ribose binds to a phosphate, which in turn is attached to another deoxyribose, which in turn is attached to another nitrogen-containing base. And so we can polymerize the nucleotides into a chain. Now, because G pairs with C, and A pairs with T, and C pairs with G, and T pairs with A, whatever it is that we generate on one strand of this polymer, there will be a complementary strand running the other way. And so base pairing leads to 2 strands, one running this way and the other one running that way, that combine together. This generates a 3-dimensional molecule that is 2 strands of polymeric nucleotides in a double helix, all stuck together with a weak attraction in the center of that helix. And that's DNA. DNA is our reserve for all of our genetic information. Those bases code for what your proteins in your entire structure, in the entirety of your body.